Okay, we'll get started. Welcome, everybody. It's my pleasure to introduce Gavin. Gavin is from Pennsylvania, and he went to college at Westminster College, where he was recruited as a star basketball player. Did very well there. He was named Student Athlete of the Year, which is a thing that basically none of us can say. But he was majoring in biology, and so he decided to get a PhD. He went to Georgetown. He worked with Stefano Vicini on NMDA receptors and how they're regulated and how they affect synaptic activity. Was very productive, published many papers, enough to convince Rick Huganier to recruit him to his lab for a postdoc at Johns Hopkins, where he continued to work on NMDA receptors, did beautiful work, was involved in a ton of projects, including one sort of perhaps more obscure project on a molecule called Syngap. He was then recruited as an assistant professor to the University of Alabama at Birmingham, where he continued to work on this gene Syngap and Syngap1, and it led to this pretty cool landmark paper in 2012 in Cell, where he basically characterized these mutant mice, the heterozygotes for the Syngap mutation, because it turns out that this gene is linked to human neurodevelopmental disorder characterized by autism, epilepsy, intellectual disability, a number of other behavioral changes. And so this was like the first time that anybody sort of had put Syngap1 on the map. And it's hard to believe that it was only 10 years ago. He's really taken this field forward. Eventually, he moved to the Scripps Research Institute in Jupiter, Florida, where he's been for 12 or 13 years. And he's continued to work on this molecule. And what I like about his work is that, he started working on characterizing synaptic deficits in these mutant mice, characterizing their behavioral deficits, but noticing that in different parts of the brain, this protein worked differently. And so he would see different deficits in different parts of the brain, which was puzzling. And he was not shy of sort of tackling this problem and trying to really dig deep and find out why is it that the protein may have different functions in different brain regions, sometimes causing hypoexcitability, sometimes hyperexcitability. And he started reintroducing the protein or the gene at different ages and rescuing deficits. So it was just very elegant work. Perhaps one of my favorite papers is one that he published in Nature Neuroscience in 2018, where he characterized tactile deficits in these mice. And whereas in a bunch of other autism models, mice tend to have this sensory hypersensitivity that correlates with circuit deficits that make sense, in these Syngap mutant mice, it's actually a hyposensitivity. And so this is standing out as kind of a different model of a neurodevelopmental disorder. In parallel, he's been working on another molecule, myosin 2B. I don't think we'll hear about that this time, but he's actually very involved in translational studies, collaborating with pharma and biotech to develop small molecules to boost protein levels in a variety of these conditions. One of the cool things that happened at UAB, besides getting interested in Syngap, is that he met his wife, who is also a neuroscientist, and I think in the majority of the papers they published together as corresponding authors. So it's sort of really like this nerdy, love affair. And so anyway, I've gotten to meet him in a meeting recently, and then we've sort of met on Zoom a few times. And what really stands out about Gavin as a person is that despite his amazing CV and all these beautiful papers that he publishes, he's sort of a very laid back, down to earth guy, refreshingly interested in science and not interested in H-index and stuff like that. Anyway, today you're going to talk about risk gene effects on cortical development and cognitive function. So welcome.
Thanks so much, Carlos. You're right, it's pretty nerdy, my relationship with my wife, but we do work very closely together. And actually, first 5 years were rough, but then we figured out how to work together, and actually in some strange ways brought us like super close. So whenever we go away to travel, it's we're always texting each other, can't wait to come back. So I'm super happy I'm here, but I can't wait to go home tomorrow. So yeah, so I mean, this is of all the places I'd want to visit, I've given seminars in seminar series around the country, around the world, but UCLA, certainly, easily in my top 5 of places I'd want to visit. So I really appreciate that. So I can my science bucket list, I could check another one off. I have science heroes in the audience. I have colleagues who I respect who've helped show me the way that, what can be done with models, and others. So, I'm just, super happy to be here. So I'm going to try to stand here and use the pointer, for those, there's, 38 people on Zoom, which is cool. Hi, Zoomers. And so, I'll just try to, do the best I can for both audiences. So, I want to start, where most people end is that, I don't really do anything in the lab anymore. I try to lead the science and raise money, and lead discussions, but all the work's done by these extremely talented people in the lab. My lab's very interested in diversity and inclusion, and I just took 3 graduate students this year, 2 females, 1 URM, and so that makes me even more proud to see that we have a diversity of faces in the lab, and they all do great work. So I'm really love my group. And just a couple disclosures: I'm a pay consultant for several biotech companies, and nothing that I'm going to talk about today has anything to do with that stuff. So, okay, so as Carlos mentions, I think my lab's going in a slightly different direction. I mean, it's a natural progression. People like Alcino and Carlos won't be surprised, but I think it's important to understand people's evolution, especially when you have an hour to give a seminar. So, really, I'm a synapse physiologist. I've studied dendritic spine biology and the molecular mechanisms that regulate AMP receptor trafficking that's related to, both synapse maturation and also LTP. And so I started my career early on. Was interested in molecular mechanisms. That's where I first got interested in Syngap, but I've also studied these molecular motors that are very highly expressed in dendritic spine, myosin 2B. And so, I'm at my heart a synapse biologist in particular, and try to understand how synapses regulate, logic of circuit connectivity. But really my lab falls on these 3 pillars, which is to understand animal models of altered synapse connectivity, which is what I'm going to talk about today. And through those endeavors, really everything that's in the lab related to biology is driven from the top down, from patient populations, genetically defined patient populations. And so we were, with the Syngap1 disorder, where we got the idea to do the modeling and sensory dysfunction, we actually got from spending a lot of time with Syngap1 kids and their families. And also there's a large part of my lab is devoted to drug discovery, early assay development, intellectual property discovery. I'm not going to talk about that today, but actually after talking to Carlos this morning, I probably should have talked about it, but maybe, I can come back in a few years, hopefully after we have some compounds that might get closer to the clinic as they relate to genetic disorders. So, okay, so that's kind of my background. And so, really today what I'm going to talk about is our desire and passion to really understand, in particular, we're interested in psychiatric disorders and behavioral disorders, but we're particularly interested in this idea that cognitive dysfunction, cognitive processing, particularly in the cortex, which maybe to a large part driving a lot of behavioral abnormalities that one sees in psychiatric disorders. And so while we study neurodevelopmental disorder genes, I think the idea is we really use this one gene in particular, Syngap1, as an entry point for understanding how genes can regulate cell biology and neuronal cell biology and create circuits that give us neural systems that can drive behaviors in cognitive function. And so the bet is that if you study the right neurodevelopmental genes and you learn a lot, you can use that knowledge to be able to understand circuit deficits, in other types of brain disorders. So that's kind of where I'm at. So I'm not particularly obsessed with neurodevelopment or Syngap1 in general. It's just that I think we keep learning enough to be able to stay, in this area. Eventually we want to take what we learn from Syngap1 and actually start looking at neurodegenerative disorders to see if we can kind of rebuild circuits and things like that. So if you just look at autism as a type of a subtype of neurodevelopmental disorder, I don't have to tell this audience the prevalence is remarkably high. It's a psychiatric disorder based on behavioral abnormalities, and so I use these arguments is that it's both socially and economically very important that we find treatments that can change the course of these disorders. It's incredibly expensive, millions of dollars per patient over a lifetime, and as a collective, it's billions of dollars, bordering on $500 billion. And there's been recent high-profile clinical trial failures, and so there's lots of reasons, lots of potential explanations for why that is. But I think one thing we can probably all agree on is that we just don't know enough about brain function to be able to surmise an etiology for, these types of brain disorders. And so, I think there's still a tremendous need for modeling the bona fide risk genes for what they do to neurons and circuits and how they affect neural systems that drive behavior. I think that will inform better translation. And so today, mostly I'm going to talk about our work in trying to really understand how Syngap1 affects the development of the cortex and how that might affect neural systems and behavior. So using autism as the entry point, we know that the symptomology of autism is really defined by a collection of symptoms defined by a clinician. And so, it's hyper or hyposensory reactivity to sensory input, repetitive behaviors, social and communication deficits, difficulties in relationships, in forming and maintaining relationships, and attachment to sameness and things like that. But it's what is the thread that permeates a lot of disorders is in peer decision making, or the inability for kids with autism or severe neurodevelopmental disorders to understand what the environment is throwing at them, process it, and then choose the right behavior that is adaptive. And so I think that concept can, explain a lot of what we know about autism at the highest level. And so I think a lot of that can be explained by cognitive deficits. And so here's an example. This is from a Syngap1 mom. I tweeted out before a meeting in October. If there was something about the Syngap1 disorder that could be mitigated, what would be the thing that as a parent you would want to see changed? Not fixed, not cured, but changed or improved. And basically here, and this parent actually is very sophisticated. She's actually in the media. And she said for her, it's cut and dry. It's cognitive impairment related to language, is that her child, while she has a low IQ, she goes to school, but she's acutely aware of the fact that she can't communicate like her peers. And it's as she gets older, it's making more and more of an effect on her, not on her relationships, but her mood, because she understands that that's one of the reasons why her relationships are struggling in school. And so from her perspective, from this parent's perspective, if we could come up with some therapy that could improve her working vocabulary, even a small amount, it could be transformational for this child. And I think that's generally the case for the way that a lot of us think about treating these disorders. Is we're not looking for cures or reversion. It's more of changing just the trajectory, just to try to help improve the quality of lives of the patients. Okay, so this is going to illustrate our general approach. Is that, like many people here, we're interested in how genes regulate, neuronal cell biology and how that influences synaptic connectivity and how those processes come together to create functional systems or networks. And so those are the closest neural substrate to behavioral control, right? And so, that's our general framework. And we have a slightly different entry point these days, which I'll get to in a second. And so while we don't really understand much about autism and neurodevelopmental disorder etiology, one thing we do know is that the cortex is disrupted, and it's largely repeated finding is altered functional connectivity across brain regions in the cortex. And so this is a review from perspective from biological psychiatry from not too long ago. And so if you take the brain has systems through this connectivity across the cortex and also subcortical areas, create what are thought as domains or systems. It depends on how you want to express it. And so from the NIMH's perspective, from RDoC, they consider 6 different domains. You know, I think that these domains should be multidimensional, right? But the idea is that these neural networks in your brain enable these different domains that can process, you know, positive and negative valence. So how does the environment affect you positively, negatively? You know, arousal and regulatory domains, social and cognitive. So the idea is to understand that the different networks in the brain are disrupted and can affect one or more of these systems, which leads to behavioral maladaptation. And so the idea would be to understand what systems are affected, and then understand how circuits that underlie and control those systems are affected, and then try to link that directly to behavioral dysregulation. So that's the approach that we take. And so I got a really good tip from David Swed, who got from Eric Kandel, which is try to tell people very early on in your talk what you're going to tell them to keep them engaged, right? And so this is basically the framework that the last 12 years has given us, and I'm going to show a lot of the evidence that underlies this. But basically, the idea that we're operating under is that why we think a lot of the behavioral dysregulation in this mouse model of this monogenic disorder, which is Syngap1 loss of function, occurs through normal sensation, but it appears that sensation, so sensory processing in the cortex degrades the animal's ability to make sense of the environment, and therefore is incapable of choosing the appropriate action or behavioral plan to adapt. And so today, what I'm going to focus on mostly is the evidence that we have of direct impairments in sensory processing directly in the cortex. But we also have projects in the lab, which I can't talk about today, but where we see there's very clear alterations as well as in the amygdala as well, for direct implications for valence processing. And we have some recent interesting evidence that the locus coeruleus may be affected, but indirectly from, we think, from improper communication with cortex. And so, so the idea is that there's normal sensation in, impaired sensory processing, abnormal behavior out. So that's just a very basic framework that we're operating under, and I'm going to show you some evidence today of that. But first, before I do that, it's important to do a quick overview of the genetics. So there's maybe a handful now of genes that, because there's been so much work done in clinical, in genomic and genetic sequencing in neurodevelopmental disorders, there's a handful of genes which are essentially causal when their mutation results in loss of function, are causal for some type of neurodevelopmental disorder with autism as a component. And you can see here. And so this is from the Autism Sequencing Consortium. This is from the Buxbaum paper from last year from Satterstrom in 2020. And you can see that the genes that are high on the list are the ones that have the highest probability of contributing risk to autism. And so from talking to the Buxbaum directly, these ones up here are essentially causal. And I can tell you that every single kid that has a loss of function Syngap1 disorder has a fairly well-defined neurodevelopmental disorder. Now, the interesting thing is a lot of the same genes pop up in completely different patient cohorts. And so this is from the DDD study from the UK, where they just looked for unexplained developmental delay. So these are kids that are 2 years old, that, where they first missed their milestones, and you see a lot of the same genes on the list. And so, and you see here, Syngap is right there. And so this was a very nice review from David Goldstein to kind of give an overall conceptual view of these different genes, is that some of these genes, when actually in the clinic, result in differential, either neurological or psychiatric diagnoses, right? And so, as we know that our brain diagnoses are human constructs, like we've invented them basically from a collection of symptoms. And so, but what these genetic studies tell us is that there are genes that underlie shared neurobiology between epilepsy, intellectual disability, and autism, is because Syngap1 is not just an autism disorder, it's also an intellectual disability disorder, and it's also an epileptic encephalopathy. And so the fact that it can lead to 3 differential diagnoses says there's something about what Syngap does in development that leads to these 3 different outcomes. And so I feel like that's a really good gene to study. So that's one of the reasons why we focus on it. A quick summary of the Syngap1 disorder. So Syngap1 disorder is essentially defined by de novo loss of function variant. So that means a variant that's not present in the parents, it's present in Syngap1 coding sequence, and results in either a frame shift or a premature stop codon. So essentially they're heterozygous knockouts for the Syngap1 gene, and we've done several iPSC studies in the lab now, and every patient, every set of neurons we've looked at that's come from patient iPSCs all have 50% protein. And so it's generally Syngap1 is a nonsense mediated decay gene. So it looks like when you have one of these loss of function mutations, you just simply lose half the protein. And so that's nice. And from a modeling perspective, is because a heterozygous knockout mouse or a heterozygous knockout neuronal cell model can actually model a very large portion of the patient population without having to individually put each patient mutation in. In terms of the clinical makeup, so here you can see that it's a loss of function disorder, loss of 50% protein. And so it's fairly common for a rare disorder. It can be up to 1% of unexplained neurodevelopmental disorder cases. That was the estimate from the global developmental delay studies from the DDD. As I mentioned, pretty much every kid has global developmental delay. Every child has epileptic encephalopathy. Nearly all qualify for an autism diagnosis at some point. I think with autism, you come in and out of diagnoses sometimes, but these kids are nonverbal and they also have some motor deficits. And so they usually qualify for pretty clear autism diagnosis. They all have intellectual disability with IQs between 50 and 60. And they have altered sensory processing, some gait abnormalities and constipation. So this is going to be a very circuit-oriented talk, but it's important to talk about what the Syngap1 gene does. And so it encodes a 135 kilodalton protein called Syngap, and it's very heavily spliced at both the N and the C, which I won't get into today. But we've recently published a paper where the different splice forms actually carry out different functions within neurons. And so that's one of the reasons why we think that this gene is a disease gene, is because one gene encodes multiple proteins that have multiple functions. And so those in the loss of function disorder, you have many different mechanisms awry at different times in development. But what Syngap, what most we know about it is it's the protein is largely expressed in brain and largely expressed in neurons and highly enriched in excitatory neurons in the forebrain. And so there, a lot of Syngap gets targeted to the postsynaptic density, where it sits within the PSD within the postsynaptic density, where it binds the PSD95 and the SAT family proteins here. And so what it does is it sits well positioned right beneath the NMDA receptor to respond to calcium to be able to initiate signaling events that are related to synapse plasticity. And it does that through its RASGAP domain. So work by Rick Huganier very elegantly showed that whenever NMDA receptors are activated and Syngap gets phosphorylated by CAM kinase, Syngap can be ejected out of the dendritic spine. It physically moves. And so what you end up with is you have a de-repression of RAS-ERK signaling, because what Syngap is a RAS-gap. It shuts down these G proteins. And so when it gets ejected from the spine, it leads to an initiation of these small molecule or of these small GTP pathways that regulate AMP receptor trafficking. Okay, so that basic mechanism, and there are other mechanisms of Syngap, but that's most what we know about. And so you can take this down into two functional. So what does this gene in protein do to cellular mechanisms? And so you can break this down into roughly two categories: altered synapse function, and particularly dendritic spine synapse function, so excitatory synapses in the forebrain. But it also has a pervasive effect on dendritic morphogenesis. And I'll show you some data at the end of the talk, which these are dissociable from synapse deficits and are entirely different mechanism. And so that's important because, of course, dendrites are the platform for which synapses can form on. And so if you have altered synapse biology, each dendritic spine synapse can kind of have its own life because we have input specificity. Whereas if you alter dendritic morphogenesis, you're kind of by definition altering neural circuit pathways, right? And so if you imagine that you have altered dendritic morphogenesis and then mapped on top of that to have synapse plasticity deficits, you can imagine that you have a problem of intrinsic wiring of the brain, and then the brain lacks the ability to modify those improperly wired circuits because plasticity is lacking. And so recently we published a paper in human neurons that come from patients and show that both altered synapse function and altered dendritic morphogenesis are both present in human neurons as well. So we think they're species conserved. So we're going to talk a lot today about how sensory processing in the cortex may be related to cognitive function and decision making, and so this all started from my work with the Syngap community, and so we organized the first international Syngap meeting. I think Alcino was in attendance. Thank you for supporting us. That back in the early days when Syngap was still obscure. And so it's from those early meetings that momentum gained to be able to create a Syngap1 patient registry, where we just signed up as many Syngap patients as we could in collaboration with Jimmy Holder and a couple of other people. He's a pediatric neurologist at Baylor, and as well as some family organizations. And so we did that because we wanted to just get a good survey. There's no clinical, there's no clinical research on Syngap. So we needed to get some information on phenotypes specific to the Syngap1 disorder. And so one thing that popped out, I got together with some psychologists, we did some questionnaires, and we just asked to, like, can you explain some, abnormal behaviors or responses of your kids? And so what really popped out were things that related to tactile sensitivity, or the lack of tactile sensitivity, in particular painful stimuli. So here you can see that, there are many anecdotal reports, and it's present in this 2018 paper, where there's some, kind of horrifying, if you're a parent, some horrifying, or just a human, horrifying descriptions of some of the challenges that these Syngap kids face. So for instance, there will be stories about how they'll, slam their fingers in a car door and no response. The parents only find out about it later on because they're, they can clearly see that the twisted fingers and black and blue fingers. Another parent told me a story where a child climbed a tree, fell out of the tree, from 20, 30 feet, kind of laid there for a little bit, dust himself off, and just climbed right back up the tree again. No crying, no complaining. I can tell you if that was my son, I wouldn't hear about it. I'd be hearing about it for 2 days, and he wouldn't climb a tree for 2 weeks. But then he would probably try again. Anyways, so, and this is just, from a parent's own words. It's just the type of how, apparent sensory abnormalities can lead to adaptive functioning is that, these kids will just repeatedly put themselves in harm. And if you read the literature, the clinical literature, nobody has any explanation for this. It's completely unknown why, kids, and this is generally across autism, whether it's a genetic form of autism or an idiopathic form, that there seems to be a lack of responsivity, in particular to very painful stimuli. In fact, there's lots of reports of caregivers for kids with autism where they believe that these kids are hyposensitive to pain in general, that they have what are called high pain threshold. And so there's a really nice article in Spectrum News about this, is that could potentially be very problematic, because in some clinical research studies, kids that improperly behaviorally respond to behavioral stimuli, if you take physiological measures, so heart rate, blood pressure, skin conductance, they can actually hyper respond. And so because this is a communication and behavioral disorder, it's intriguing to think that one of the issues could be is that the brain, and particularly the cortex, has a hard time making sense of the stimuli and then telling the action centers of the brain that create behaviors to pick the right behavioral repertoire to be able to defend themselves. And so that's kind of where this all led to, is just the lack of satisfaction of reading the literature to understand how a whole population of kids with different genetic etiologies can have this lack of responsivity to pain, and so or tactile stimuli. And so that's obviously not the case. I mean, there are clearly instances of kids that are hypersensitive to tactile stimulation, but in general, and even with that, there's a very poor understanding. And so just generally trying to understand how the brain takes sensation, transforms it into sensory information to behave, I think is an important area to study in autism in general. And so that's the driving force behind this. And so we sat down and tried to understand what would be the best system if you're going to do this in an animal model. And so this is where one of the areas where animal models are super important, because we want to understand neural systems. So how does the sensory in lead to behavior out? We need an intact animal for this. We can't intervene in humans. Mouse has the most sophisticated genetic tools. And so then within a mouse, we asked, so what are the systems in mouse where we can use Syngap model mice, and to then ask and then create behaviors where they have to make decisions, and then be able to have an understanding of the circuits that underlie those behaviors. And so some of the amazing work has been done in the mouse tactile system, especially in the Whisker system, where people like Carl Peterson and Dan O'Connor and many other people have helped us understand exactly what the circuitry is that underlies the mouse whisker system, which is their principal touch organ, and how that leads to adaptive behaviors. And so we thought that would be a good intersection to take these genetic models and then work on this system. And so one of the things that this field has given us is this idea of ascending loops for different levels of processing. And so you have a sensory brainstem motor loop, which is for reflexes, right? No decision making whatsoever. So you need these very quick loops with just a couple synapses to be able to defend yourself quickly. But then you have ascending loops where you have peripheral to thalamic loops. And then you have brainstem to cortex loops. You have cortical thalamic loops, and the idea is that these ascending loops allow for more and more sophisticated processing of the sensory stimulus to be able to give more and more meaning to it. And that's precisely how sensory perception emerges. Is the sensation is converted into a sensory stimulus with meaning, so then the brain can figure out which behavior to generate to be able to undertake the most adaptive behavior. Okay, so as I mentioned, if we want to take this approach, the idea would be to try to understand functional systems. That's going to be the mouse touch system by Whiskers. And then we want to get some evidence in the Syngap model that there's some alteration in behavioral control that's due to alterations in the mouse whisker system. And so this part of the talk will be intermixing of previously published work and new work. So this is some unpublished data that we're getting ready to submit for publication, which is high-speed videography of a mouse trying to discriminate two identical objects that differ only by texture. And these are very subtly different textures. So these would be the difference of about 1 millimeter width in the grooves. So as I mentioned, they're identical in every way. And so a wild-type mouse does a very, very good job over here. This would be the discrimination index. So this would be how well can it discriminate the original object, which has a certain texture, versus a new object, which has a slightly different texture. Mice, like humans or other mammals, will spend more time around a novel object. And so whenever they spend more time around the new object as a different texture, we assume that the animal was able to discriminate. And so when the animal is able to discriminate this in wild type, when you trim their whiskers, they have no more discrimination. So we know it's whisker dependent. So in the Syngap1 mice, not surprisingly, based on the content of the talk, is that Syngap mice fail very miserably at this. However, so they have no discrimination whatsoever, but it could be that they just have problems expressing novelty. They may have short-term memory problems. And so what we did is we looked at basic novel object recognition. So it's the same task, identical arena. The only difference is instead of having two identical objects that differ by texture, we just had two slightly different objects, right? So a typical novel object recognition task, in this case, a master lock and a stapler. And in this task, Syngap mice do this very well. They have no problems doing that. So that tells us that novelty expression is normal. It tells us that short-term memory is normal. It tells us that there probably is something specific to the tactile domain that prevents them from being able to understand the two objects. And so we were pretty excited about this talk. I'm very excited about this talk. We were very excited about that data. And so then we asked ourselves, well, we wanted to start to extract mechanism. And because of these beautiful papers from David Ginty's lab, where a lot of the sensory abnormalities that are in several different autism models can originate in the periphery, we knew from the expression profile of Syngap, it's not really very well expressed in the periphery. And it's actually not very well expressed even in subcortical areas. It's very highly expressed in excitatory neurons in the cortex. And so we thought that, well, this might be a good model for us to understand cortical dysfunction as it relates to sensory processing. And so here's some data that we mined from the Allen brain, that's on their database for single cell sequencing. And here you can see a very nice demonstration we recently published in an eLife paper. The Syngap is indeed highly expressed in various different types of forebrain glutamatergic neurons. Probably most highly expressed in layer 4 or 5 and layer 2, 3, cortical projecting neurons. Interestingly, there is expression in GABAergic neurons, but it's much less. And there's very little expression in glia, which is also found in culture neurons. So this told us that maybe we should regulate Syngap's expression in these cortical excitatory neurons, and perhaps that can explain the origins of these tactile deficits. And so we have several models that I'll introduce you to. So we have rescue lines. So it's a heterozygous knockout mouse, which genetically models the patient population. And then when CRE is expressed, it reactivates the destroyed allele. So you go from heterozygous animal back to wild type wherever CRE is expressed. And so EMX1 CRE line expresses CRE in forebrain excitatory in glutamatergic neurons. And so we cross those two lines together. So the whole entire mouse, whole body, whole brain is heterozygous except for cortical excitatory neurons. And in this case, we can see that the discrimination comes back. So So then we wanted to get more evidence. So we tried to do more sophisticated operant behaviors that are dependent upon whisker stimulation. And so here's a variant of a quasi go no go task where the animal is head fixed. And this is a lick port. And then so the animal has to learn over time that when you stimulate the whisker, he's supposed to lick the sensor to get his water reward. He's very thirsty. Okay. And so over time, the animal doesn't know the rule, but then eventually figures out that if he licks the sensor when the stimulus comes, he's going to get his reward. And so what it becomes is a sensory detection task. And so that's a form of learning over time. And then we can also test the sensitivity, to how much sensory stimulus do we need for the animal to perform. So it's for two reasons. It's a really good task. And this is just an example of it here. You'll see the piezo will go, and then the animal licks the sensor, and then the animal will do 200 trials per day. We can do 12 animals. We can do 16 animals per day, and we train them over the course of 21 days. And so this is a wild type mice, okay? And so this would be a cortical knockout mouse here. So the conventional hets look basically identical to this. I just didn't show it for time. So we can recapitulate. So they have a learning deficit. So this is the fraction of correct trials over session days. And so wild type mice, start pretty low and then learn, relatively quickly, whereas the Syngap mice do learn, but they learn much slower and they never quite get to the performance of the wild types. And again, this is a selective heterozygous mouse in the EMX1 population. So the whole body and brain is wild type except for cortical glutamatergic neurons. So then we have this rescue line that I showed you before. And so interestingly here, this prevents the behavioral deficit. So here in this case, the whole animal is heterozygous except for cortical excitatory neurons. And so here, we get absolutely no phenotype. So it looks like that this in another type of sensory detection task, a tactile detection task, that it looks to be the origins of it appears to be Syngap's function within these cortical glutamatergic neurons. And so these were exciting to us because now it gives us, being a neurobiologist, now we have a defined population of cells. So what's going on with these neurons and how can this relate to what we're seeing behaviorally? And so here it looks like there's a problem with the cortical loops where we think that generally meaning is given to the stimulus so the animal could behave. Okay, so I think I was giving a talk to a much less sophisticated audience, so that's why there's a lot of other things. Okay, so as I mentioned, now we have the gene, we have the system, we have behavioral control problems, and so now we want to get into the nitty gritty. Is that what's going on under the hood? What's the problem between the neuronal cell biology and the synapse connectivity that leads to circuits? And so again, the Whisker system is phenomenal for this, is because there's so much known about how signals during touch or during whisker stimulation travel through various areas of their brain and get into the cortex, and in particular within what's called the barrel cortex, which is where an area of the brain that receives a direct connection from the thalamus into layer 4 and layer 5 of the somatosensory cortex, where this touch information comes in and converges. And in fact, you can find a particular whisker has a sensitive area within the brain, so it becomes very straightforward to be able to study the function in the cortex of a peripheral stimulation. It's pretty easy to find sensitive areas and sensitive neurons. And so this would be the map of the circuits coming into the cortex. And so the first obvious thing to do would be to study what the functional outcome of a whisker stimulation in this mouse where we think that it's a cortical deficit. And so we use a stimulus paradigm where we just use a piezo to be able to stimulate this whisker. And then we use the various types of imaging methodologies to look at this. One was to just do what's called wide field Gcamp imaging. So these animals express Gcamp throughout the whole brain. In this model, we use a GP 4.3 mice. So it's Gcamp's only expressed in excitatory pyramidal neurons. And so we did this whisker stimulation, and we find that over time, there's a nice activation of this whisker sensitive area, and that's it's traveling probably to other areas of the cortex. But in Syngap heterozygous mice, we were very surprised to see that there's very little activation. And so we got these data way back in 2015, and we were shocked by this because everything we'd ever seen in this mouse up to this point was hyperexcitability. Cortex or prefrontal cortex, hippocampus, you name it, hyperexcitable. Here we're seeing, wow, we have a hard time even detecting signals sometimes. Then we went in and then tried to understand the cellular mechanisms of this. We did Gcamp imaging. So it would be to look at individual neuronal activations within these sensitive areas. And so here we do a series of whisker stimulations. That would be each line. Each number is a different active neuron. Green asterisks represent neurons that respond to the stimuli. Red asterisks represent what we would call, kind of ongoing activity or non-stimulus dependent activity. And so what we found is that there was perfectly, so this would be wild type, this would be heterozygous mice. And this is in the EMX1 Cree model, by the way. We did this in the conventional mice as well, but I want to show, I'm trying to emphasize the cortical deficits here. So what we found is that there's apparently normal ongoing activity or non-stimulus dependent activity, but there was much less stimulus dependent activity. And so we think that that's the explanation for why we see less of this wide field imaging that we see in the cortex. And so this is just one way to look at this. This is response probability. So what's the probability that a neuron will respond to a given number of sensory stimuli? And this would be the cumulative fraction. And you can see here that there are populations of neurons that should respond almost all the time to sensory stimuli. And these types of neurons are essentially gone in Syngap mice. These highly active neurons that respond over and over again are just not present. And so that might be perhaps one of the explanations for why they have a hard time discerning these tactile tasks. So because that was in layer 2-3, because that's the most accessible population of 2-photon imaging. The wide field imaging is, no one really knows, it's surface of the cortex. So it's an integration of lots of different types of neurons. So then we wanted to do a probe. So then we wanted to get a little more sophisticated to get more information. Number one is that we wanted to look at naturalistic touches. So the previous experiment I showed you is artificial whisker stimulation. So the animal's head fixed, and we artificially move the whisker ourselves to be able to just extract as like kind of an evoked sensation. So in this task, I don't know if I have it in. So yeah, so basically what we did here is we did two things. One, we head fixed the animal, but we let him explore an object with his whisker. And so this is just a pole. It's completely novel. It's in the dark, so the animal doesn't know it's there. And so when he or she, because we use both sexes, touches the object, the whisker bends, which activates Merkel cells in the face pad, which then results in action potentials that set throughout the entire whisker system. And here, what we're doing is we put a silicon probe in the somatosensory cortex, and so we can see that every time the animal touches the pole, you can see very nice activation in this neuron in S1 cortex. And so what we did in this experiment is we did 12 animals per genotype. And we put probes in the same animals in S1, in the somatosensory cortex, in the motor cortex, because that's the loop goes from somatosensory to motor and then back down to the thalamus. And so we put the third probe in the whisker thalamus. And so the idea here is that by using these probes, we can get a better understanding of the major components of this top-down loop that processes first sensory information in S1, but then routes it to other areas of the cortex. Today, I could only show you data from the S1 probe, but the S1 probe is, we're very comfortable with it and we think it's ready to publish. We are currently looking at M1 in the thalamus, but I can tell you whatever I'm going to show you in S1, nothing like that is happening in M1. We're trying to actually figure out what it is, but it is definitely not the same signature that we see in S1, which we think is super cool. So this line, so this is we did, so the cool thing about this is that these are spontaneous touches, so we can get more than 500 touches per animal, and across lots of animals, we have lots of power. And so we can get really nice activation. We can look at very nice neural activation curves. And so, each animal, we can get, I think dozens of active neurons. And so we have more than 10 animals per genotype. So we're talking, maybe more than 100 neurons per genotype here easily. And so what we find is if you just look at the S1 probe, you get much less activation during naturalistic touches in the heterozygous mice, which is in red versus blue. And that fits very well with our Gcamp imaging that was, through kind of a non-naturalistic stimulation. And so we're quite convinced that there is really less activity generated in the somatosensory cortex during touch. And so the nice thing about silicon probes is, as we were talking about this morning, is that you can get spatial information as well as temporal information. And so these probes will go down through all layers of the cortex and then even down into other subcortical areas. And so what Tom did in the lab was to then look and do depth coding. So this would be layer 1, this would be layer 2, 3, this would be layer 4, this would be layer 5. This would be one animal's responses to lots of different touches, or one neuron's responses to lots of one animal's responses to lots of different touches. Each bin going across are different neurons. And so what we can see is that you get this normal activation of layer 4, layer 2, 3, and layer 5. And so the idea is that the stimulus comes in and activates layer 4 and to some extent layer 5. And then information is then routed up to layer 2, 3, and then back down out to layer 5. When we look at all of the different active units, which is in the pale colors, and then the grand average for each animal is in blue, what we see is that we see relatively normal, middle layer, which we think is layer 4. We saw much less in layer 2, 3, and layer 5. And so then when we looked at this, on the left, these 3 panels here are if we just took, so the way we extract units is the animal is in the box, he's awake for 2 hours. And so we use an algorithm to extract regularly spiking units across 2 hours. And then from those units, we ask, okay, what happens during the moment of touch? And when we do that, we see deficits in all 3 layers. However, I think you can see here that the first phenotype that we found is not only is there less activation, but there's just fewer active units in these mice. So there seems to be just less neurons activated in the ones. But the question is, the neurons that are activated, are they actually less active? And so when we do that, we took only the responsive units over here. And interestingly, what we found is now layer 4 is normal. So layer 4 is responding normal, and that's the area where the sensory information comes into cortex from the periphery. But we see reduced activity both in layer 3 and layer 5. And so another thing that we're fairly confident in is look at, so the temporal dynamics are I think something to pay attention to here. And so in layer 4, so what you normally see in layer 4, layer 2, and layer 5 is if you look in the blue traces as wild type, you see a fairly nice upward trajectory of neural firing rates. There's very little variance here, right? So you can see that the shaded blue, there's very little variance in the onset of the activation of these neurons. However, in layer 4, we see about the same level of activation in the same temporal component. But both in layer 2, 3, and layer 5, we see very different temporal onset. There's very clearly something going on drastically different in layer 5. And when we look through all of the active units, you can see that there are some units that are activated and delayed by some time. Yeah.
Gavin, is that anticipatory response before the stimulus real there? Lower layer 5, layer 6 to the right. Yeah. Is these periodic stimuli or the?
No, these are, that's the animal is free to touch when he wants. He's in the dark. So he's whisking on a pole, basically.
So that anticipatory going up, there's more anticipation in
here.
Bottom right.
Bottom right.
The red trace is earlier. Yeah.
So all I can tell you is at this point is that we are confident in the neural activity data. We don't understand it at all, and so one of the reasons why I'm showing it today is because of the people that are in the audience. So we are not systems biologists. I don't ever want to become a systems neuroscientist. We just need to look at systems biology to be able to understand the circuitry underneath. So, that is, I just want to be very clear that we're not going to turn into a systems neuroscience lab. And so we're going to need some help to understand this. But some data that I can't show you today is we did rabies tracing for the loops in the cortex, so S1, M1 connectivity. And so we looked at afferent connectivity into S1 layer 5 neurons, okay, using retrograde rabies tracing. And we did whole brain mapping, okay? And so everything is completely normal onto those layer 5 neurons except motor cortex. Motor cortex is very highly hyperconnected in Syngap het mice from M1 back to S1. And then when we ping that with optogenetics, it's 3 or 4 times hyperfunctionally connected. And so the theory is, and this is just a theory, is that there's some sort of kind of circuit level homeostasis happening, that these layer 5 neurons want to spike a certain amount, right? They are engaging in compensation circuit wide. And so we think that the reason why there's an M1 to S1 selective hyperfunction is we think that one thing we know about this loop is that S1 goes to M1 and then M1 comes back to S1 and then S1's fire out. So an intriguing hypothesis would be that there's some delayed response coming from some other brain region to try to boost up this deficit in signal, and that's one of the things we're considering looking into. But I think what it is it's a signature of altered functional circuits within these somatomotor areas, and so I think it's less about the valence. And well, it's equally important, the amount of reduced spiking, but also the temporal aspect, I think, is super important as well, based on my reading. So,
Can I ask a little question?
Sure.
Did you talk about Whisker trajectory, where in ways I could imagine one of the cases could be that these mice behave differently?
Great point. So there's absolutely no difference in any of the probes during whisking. So this is a 2-hour-long experiment. Thanks for pointing that out. We even track whisker behavior, and so we looked at whisker bending and all that stuff. There is and Alcina saw some of that in Fiji a few years ago. So before the pole comes in, they have a whole period in the dark where they're just free whisking, and there's no change in the dynamic of the whisker, and there's no change in neural activity. So all of these are normal. The only difference is in M1, there's actually hyperactivity in Syngap het mice. But there's no change in temporal dynamics at all. It's a little bit of difference in valence. So whisking dynamic looks normal, and activity during whisking looks normal. So like kind of the arousal, motor efference, copy stuff, that all looks normal. Interestingly, when we track their whisker behavior, as soon as the pole comes in, Syngap might start using their whisker in all kinds of very odd ways. When we did this touch analysis, we had to get rid of all those odd touches. So we're actually looking for the first touch in a series. So they're pretty much apples to apples between animals. And so that's why we think we get this normal activation in layer 4, because we're looking at kind of stereotype touches. But if you look across all touches, I mean, it's really messed up, because these Syngap mice kind of like ram their whisker into the pole and don't sit there and try to explore it like a wild type mouse does. We think because they're cortically blind to it, we think that they don't know it's there, but that's a theory. So, okay, I'm almost done. So there's only a couple slides left. Yeah.
I have a question. Is this cumulative for all the whiskers or only for?
We've only checked with one whisker so far. We're doing pole localization tasks where we look at 3 whiskers, to get at this. So so it's a good question. And we're also going to do some neuropixels with air puffs as well in development to get at this. So yeah. All right. So as I lied, I have a bunch of slides, but I'm going to go through it quickly. So I want to get at the mechanism. So we have a whole nother study on the mechanism. So what is Syngap doing inside of somatosensory cortex excitatory neurons to prevent excitation, right? And to make a long story short, we have a whole study where we figured this out. And so what it does is that Syngap essentially, we published this in 2018, is that in adult mice, layer 2-3 neurons are super tiny. Layer 2-3 excitatory neurons are super tiny. They have shorter dendrites, and those dendrites that are there have less dendritic spines, okay? We confirm this by EM. There's just less spines, excitation layer 2-3, okay? So, and we show that affects the way layer 4 can signal to layer 2-3. All right, and we show that this is postnatal day 10. So this small neuron phenotype is present very early in development. And so we wanted to understand how this happens, and that's layer 4 to layer 2-3 connectivity. Basically, I'm going to have to go through this quickly because I don't want to get the hook. But what we found is that a completely new function of Syngap. So there's something that it does that's unrelated to synapses, which is to regulate intrinsic membrane excitability. Not surprising, most autism genes regulate synapses and they regulate intrinsic membrane excitability. Syngap does the same thing. And so what we found is these neurons in layer 2-3 are initially they have very low intrinsic membrane excitability. So they have low synaptic excitability, they have low intrinsic membrane excitability, but which is cause and which is effect? We did a bunch of experiments to show that the cause of the low synaptic excitability and the connectivity is because that they have persistently low intrinsic membrane excitability during the period of development where neuronal spikes are used by these neurons to grow the dendrites. That's a very nice paper from Mu-Ming Pu's lab showing that if you artificially suppressed intrinsic membrane excitability in developing somatosensory cortex neurons, these neurons become stuck in this small state because they need a certain amount of spikes for trophic growth. And so, so we read that paper, we saw this result, we're like, well, maybe this reduced intrinsic membrane excitability is just preventing less spiking during development, and that's why we have these neurons. So what we did was then did some current clamp experiments to figure this out, and basically what we found is that these neurons in layer 2-3 are both hyperpolarized, but have reduced input resistance. That screamed like open potassium channels to us, right? So then when we look and then we investigated it and we measure potassium channel currents directly, we found that there's indeed an enhancement of potassium channel function on the surface of these neurons, which explains both hyperpolarization and reduced input resistance, okay? And so we asked the question then, well, so because of the Mu-Ming Pu paper, so if these neurons have less intrinsic membrane excitability, if we were somehow able to bring the intrinsic membrane excitability back up to wild type levels, would that then restore the dendritic morphogenesis and bring the connectivity back in the cortex? So we did this using a very cool tool. So dominant negative KV4.2 subunits are awesome because they integrate with lots of different potassium channels, and what they do is prevent them from getting to the membrane. So it's a really great way to just sequester potassium channels off the surface. And so, so we did this, and to make a long story short, we did this by in utero electroporation E15. So we're targeting these Syngap neurons in the embryonic stage and through postnatal. We measured it at postnatal day 14. And to make a long story short, this tool works. It increases intrinsic membrane excitability in both wild type and heterozygous neurons. So then we spent, a lot of time. This is the last slide, basically, in showing that indeed, when we take neurons from Syngap heterozygous mice that are either minus or plus for the potassium channel, because these neurons are right next to each other, only a small percentage of neurons actually are transfected, so we can compare pairs. And what we found is that we got a significant increase in the dendritic morphogenesis. We brought mini frequency back, which is in the 2018 paper. We showed that mini frequency was a proxy for neural connectivity. So we brought mini frequency back, and most importantly, we brought back layer 4 to layer 2-3 connectivity back. So, so we see here that we can rescue the layer 4, layer 2-3 connectivity back. So, so the model that we have for this is that in developing the layer 2-3 somatosensory cortex neurons, you have to have a balance of potassium channels, calcium channels, sodium channels, right? And you have a certain, nature gave these neurons a certain amount of intrinsic membrane excitability. The interesting thing is RAS-ERK signaling is already related to potassium channel trafficking, and Syngap dysregulates RAS. And so what we think happens is that you have an excess of potassium channels on the surface because you don't have enough Syngap there, and RAS-ERK is disrupted. And what that does is prevent spiking through this 2 or 3 week period of development, where we know that these neurons need spikes to grow. And so by suppressing these potassium channels, we give these spikes back to the neurons, and then we can extend the dendrites and enable connectivity. Now, I want to add that not only do these neurons have less synaptic connectivity because of this reduced dendritic morphogenesis phenotype, but they're also persistently have reduced intrinsic membrane excitability, even into adulthood. So this is a double hit to these cells. They have less synaptic feed-forward excitation coming from layer 4, but when that synaptic excitation arrives to layer 2-3, these neurons are sitting there hyperpolarized and have reduced intrinsic membrane excitability. So, it's not surprising then in this framework that we see much less spiking in somatosensory cortex principal neurons. And so that's kind of how we put it into a circuit framework. I was going to show you this to blow your minds, but I'm not going to say. All I can tell you is that this, if people always ask you, what about other brain regions? This happens also in every cortical neuron we look at, and they fall into 2 categories. Sensory cortex, we see exactly the same stuff in visual cortex, and auditory cortex we see in somatosensory cortex. When we look at any associational area, prefrontal, retrosplenial, so far, we see exactly the opposite phenotype. It's cell autonomous, it's in the opposite direction. So prefrontal neurons in development are larger, have enhanced membrane excitability, right? And so, and instead it's cell autonomous, same gene, same layer 2-3 neuron, different area of the brain, completely inverted effect on the way that they grow and regulate potassium channels. So too many potassium channels in one area, too little in the other. So because of that, I'm very bearish on the idea of using neurophysiological tools to be able to correct circuit deficits, because if you boost up somatosensory cortex function, you're going to make these other associational areas even more hyperexcitable. And so the idea is either targeted delivery to brain areas or put the protein back in, which is what we do in our lab. So I just like to say thanks to everybody. And there should be a thank you slide here for everybody again. And I'd like to thank our collaborators and the funders. Thanks a lot, everybody.